Hey everybody, back here in the lab, and we are going to be going over our next set of tests, biochemical tests. So we're going to continue on with our metabolism. So the selective and differential media, all of those, we're looking at different types of metabolism and which bacteria were fermenting, which ones weren't. And we're going to continue on with that with the carbohydrate fermentation section. So this is the TSI and carbohydrates together. So I'm going to go over the carbohydrate fermentation first. Then we're going to go over the triple sugar iron, which is the TSI. Then I'm going to show you how to inoculate those. And then we are going to go over the results because I actually have some that are ready to be read. So starting out, let's talk about the title of this exercise, carbohydrate fermentation. So we are going to be looking to see which carbs or sugars can the bacteria ferment. So it's kind of similar to the selective and differential media, but for those we were only looking at one kind of sugar, mannitol or lactose. Okay? So these we're going to be looking at 4 different sugars. Now, there are other sugars out there that can be added to this test. It's basically you just go down the list and there's a whole list of sugars and you say yes or no whether your bacteria can ferment those sugars or not. That helps us to identify what kind of bacteria we have based on which sugars it can ferment. But we're only going to do 4 in this class. So I've set up kind of a little mini chart. Looks kind of similar to the ones for the selective and differential media. It's just way less complicated. So our substrate for these carbs are 4 different ones. We have 4 different sugars: lactose, glucose, sucrose, and mannitol. So those are the 4 carbs that we're going to be testing in our lab. We have 2 disaccharides and 2 monosaccharides. The 2 monosaccharides are glucose and mannitol. Glucose, of course, being the one that pretty much everybody remembers. And then mannitol is a sugar alcohol. So that's that OL that you see on the end of that one. And then our 2 disaccharides are lactose and sucrose. So them being 2 monosaccharides put together. So we have some monosaccharides and we have some disaccharides. So we are going to see which of these sugars the bacteria can ferment. The indicator that's in the media is called Phenol Red. So this should sound familiar to you because you have seen it before. It is the indicator that's in the MSA plates. So, just like the MSA plates, these tubes start out red because that's the phenol red that's in the tube. So here's my tube. Okay, so it starts out red, just like the MSA plates. So, since we're looking at fermentation, we are going to be looking for acidic products. So when the bacteria ferments the sugar, it produces acid as the end product. So when acids are produced, the pH is going to go down and the phenol red is going to turn yellow, just like in the MSA plates. So that is a positive reaction for fermentation. That tells you that that bacteria can ferment that sugar. If you get a red reaction, so it stays this color red, sometimes it'll turn an even darker red, that means that the bacteria did not ferment the sugar. It used it for respiration instead. So these are going to be bacteria that are going to be doing respiration. So the ones that produce the red color. So they are using oxygen as a terminal electron acceptor, and they have an electron transport chain, and that's what they're doing. They can't ferment the sugar. They don't have the enzymes that are responsible for fermenting that specific sugar if you get a red reaction. So it's as simple as that. That's all there is to it. You get a yellow tube that's positive for fermentation. That bacteria can ferment that sugar. You get a red tube, negative for fermentation. That bacteria cannot ferment that sugar. Now, you'll notice that there was something else in the tube. So you can see that little glass tube that's on the inside of there. That is called a Durham tube. So it's basically a mini test tube that's turned upside down and placed inside of the test tube. And that's done before we put the media into the tube. And that is there to catch gas. Some bacteria, when they ferment, can produce gas as a byproduct. Some can, some can't. It depends on which bacteria you're dealing with. So that's giving us more information about the bacteria. So when we're reading these, we got to read the color first to see if fermentation occurred. Then, if you got a yellow tube, you need to look for gas production because you can have a positive reaction, a yellow tube, with a gas bubble or without a gas bubble. So, we're going to go over that whenever we read these results. So, that's it for the carbohydrate fermentation tubes. Now, we're going to move on to the TSI or the triple sugar iron. This test is done with the carbohydrate fermentation because it's looking at the same stuff. So, This is a TSI tube, so it's a slant, and you'll notice that it's also red. So let's talk about what's in this media. It's called triple sugar iron. So that means it's going to have 3 sugars in it, and it also has iron in it. It also has other things too, but those are the things we're going to focus on right now. So it's got 3 sugars in it. Which sugars does it have? It has glucose, lactose, and sucrose. So it doesn't have any mannitol, but it has these 3. Also, you will notice that they are at different concentrations in the tube. Glucose, which is our monosaccharide, small sugar, is at 0.1%. Lactose, a disaccharide, is at 1%. And sucrose, also a disaccharide, also at 1%. So that's where the name triple sugar comes from. There's also phenol red in the tube. So what are we going to be looking at? Fermentation. Can the bacteria ferment these sugars? Yes or no? Now, since this is a slant, the way that we're going to read this is called slant over butt. So these tubes, the carbohydrate fermentation tubes, are liquid. So the whole tube's going to turn yellow or the whole tube's going to turn red. Since this is a slant and these sugars are at different concentrations, you could potentially get different colors in the same tube. So let's talk about those colors that we're going to get. If you get an all yellow tube, so the entire tube has turned yellow, that means that fermentation has occurred, and you are going to record a reaction of A over A for that tube. If you get an all yellow tube, A stands for acid. So if you get an all yellow tube, acid was produced in that tube, you get an A over A. Both the slant and the butt have turned yellow. So that is a positive fermentation. If you get a half red and half yellow tube, so the slant of the tube is red and the butt of the tube is yellow, that would be K over A because K stands for alkaline. Remember what I said. If you get a red tube, that means that respiration happened, not fermentation. So you're going to get those basic or neutral end products. Another word for that is alkaline. So a red reaction, you would have to put a K there. So you get a red slant and a yellow butt. Your answer is going to be K over A. So what that means is that some fermentation occurred. Because if the tube turned yellow at all, it's yellow because fermentation occurred. And then you can get a K over K tube, which means that no fermentation occurred. So the bacteria did not ferment any of these sugars at all. So the K over K is pretty straightforward. You get an all red tube, no fermentation happens, can't ferment glucose, lactose, or sucrose. Now that doesn't mean that it can ferment, it might be able to ferment other sugars, but it can't ferment these. So that's easy. All red, K over K. Now, let's talk about K over A. So I've got the butt of the tube is yellow, so that means that some fermentation happened. Well, we've got 3 sugars here. How do we know which one it fermented? Well, let's talk about the concentrations. Remember, glucose, which is a small sugar, is only at 0.1%. Lactose, which is a disaccharide, it's larger than glucose, is at a 1% concentration. And sucrose, also a disaccharide, is at a 1% concentration. So, which number is smaller? 0.1. So, if only the butt of the tube has turned yellow, then that tells us that glucose was the only one that was fermented because only a small amount of acid has been produced. A small amount, we're dealing with a monosaccharide, and it's only at 0.1%. So only a small amount has been produced, so only the half of the tube turns yellow. Now, let's talk about A over A. That's an all yellow tube. So the whole thing has turned yellow. Now, if you get an all yellow tube, there is no way for us to know which one of these it fermented. Did it ferment just the lactose, just the sucrose, all 3 of them, 2 of them? There really is no way for us to know. So, to differentiate which one it fermented, we do this. That's why they are done together. We do the TSI. Depending on what reaction we get here, we're going to go up here and then look at these and see which ones it actually fermented. We break the sugars up. And that's why these are always done together. Now, let's talk about the iron part of the media. So we've already went over the sugar. We've already went over the fermentation. So let's talk about the iron part. There is also in this media an amino acid called cysteine. Some bacteria can deaminate the cysteine. When that happens, okay, they're breaking down the cysteine. When that happens, hydrogen sulfide is produced. Very similar. This reaction is very similar to the reaction happening in HEA. Remember we had to look for H2s in that? This is a very similar reaction. So, when H2s is produced, it reacts with the iron in the media and produces iron sulfide. That produces a black butt. So the tube will turn black. You will see black on the in the tube. Okay, and so that's positive for H2s. So triple sugar iron is a lot to read. Okay, you've got to look at the color, slant over butt. You've got to look for the H2s production, but also remember, in the carbohydrate fermentation tubes, we had that Durham tube that's in there. That's there to catch gas. Well, in the TSI tubes, if the bacteria produces gas when it ferments, it's still producing gas. So even since it's an auger, it's a slant, when the gas is produced, it creates cracks in the media. And you're going to see that whenever we read the results. So triple sugar iron, you got to basically look at 3 things. Number 1, we've got to look at the color. Read slant over butt. Did it ferment, yes or no? If it fermented, did it produce cracks in the media? Does it produce gas? And then lastly, the H2s, hydrogen sulfide production. Is there black on the slant? If there's black in the slant, that's positive for H2s production. Okay. Then once we read our triple sugar iron, we go up to the individual tubes and we break it up to see exactly which sugars it ferments. So let's move on to the inoculation process. Here's what a carbohydrate fermentation setup looks like. So we've got our TSI slant, and we've got our 4 sugars. Now, you'll notice that some of the sugars, they're not all the same color red, all right? Some of them are a little lighter orangey looking color than the bright red, but they're still all a form of red. So that's what we're looking for, okay? So we've got our TSI, we've got our lactose, glucose, sucrose, and mannitol. Now, you don't have to worry about the colors of the caps, okay? You don't have to memorize that. You don't have to worry about that. That's all in your lab manual under the procedure. It'll tell you which sugars are which cap color. But I always just try to match the order of which I put it on the board. So that's the order that I have it up on the board: lactose, glucose, sucrose, and mannitol. So that's how I always like to keep it. Okay. So to inoculate these tubes, now I'm not actually going to inoculate them, but I'm just going to show you how I would do it if I was doing it in class. I would use my loop, flame my loop, go in and get my bacteria. You could use it off of a slant. And to inoculate the TSI, I would go all the way down to the bottom, stab all the way down to the bottom of the tube, and come back up and slide across the top of the slant. You want to make sure to inoculate the entire tube all the way down to the bottom. Because remember, we're reading slant over butt. So if you don't stab all the way down into the bottom of the tube, you're not going to get the bacteria all the way down there, and it can potentially mess up your results. So you want to make sure you stab all the way down and come across the top of the slant. I would flame my loop again, go in and get more bacteria. Now, for the tubes, the liquid, all you do is dip and swirl. Flame my loop again, go in and get more bacteria, and I would do that for every single tube. Okay? And of course, I would have a bacteria per row. So, I have 3 different bacteria that I did for the carbs and TSI. I did E. coli, Citrobacter frundi, and Pseudomonas. So those are the 3 bacteria that I inoculated for carbs. So let's start out by reading the E. coli tubes. So we'll start out with our TSI. So remember, the first part that we have to read for the TSI is the slant over butt. So what color is the tube? So as you can see, my tube is all yellow. Remember, it started out this color. It's all yellow. So for my E. coli, my result would be A over A, an all yellow tube. So E. coli fermented these sugars in the TSI. Now we don't know which ones. We got to break it up and see which ones, but it fermented something in there. Now, you will see that there are huge cracks. This is awesome. Huge pockets of air in there. They have CO2. Look at that huge pocket right there. Look at all those cracks in that media. Okay? So E. coli produces gas when it ferments. This is a huge positive result. Okay? I'm so glad that we had great results for that. And the last part is the H2s production. Do we see any black in the tube? There is not any black in that tube at all. Okay, so it would be negative for H2s. So if you were recording this, you would record A over A with gas, no H2s. That's how you would record it for E. coli. So now, since we got an all yellow tube, we need to break up the sugars and see which ones it fermented. So first is going to be our lactose. So it's yellow, so that's positive for fermentation, and you see the gas bubble in that Durham tube. There, so that's a positive reaction for fermentation with a gas bubble for lactose, which we already knew E. coli was a lactose fermenter because of the selective and differential media, but we did it anyway. Here's my glucose tube. Yellow, so that's positive for fermentation with a gas bubble. So positive with gas. Sucrose, which is the yellow cap, that is a negative reaction. So that is a red tube, negative reaction. So in our TSI, remember the 3 sugars that were there, glucose, lactose, and sucrose. Well, it can ferment the lactose and the glucose, but it clearly cannot ferment sucrose. So E. coli does not have the ability to ferment sucrose. And lastly, our mannitol. Nice bright yellow tube, and look at that big old gas bubble. So it's positive for gas. So positive for fermentation, positive for gas. So the pattern that E. coli does for our 4 sugars is positive, positive, negative, positive, with all the positives having gas. So that's E. coli. Now let's move on to Citrobacter frundi. So the first one we're going to read is our TSI tube. Looks very, very similar to our E. coli. A over A, lots of gas being produced, and it's negative for H2S. Now, normally Citrobacter frundi would give you black colonies, and if I let it go for a little longer, it might give us some black colony, a black test tube, but it's not today. So normally you would get black H2S production for that Citrobacter frundi. Okay, but you can see there's a little bit of black starting to form inside of the tube, but it's going to be kind of hard for y'all to see. Okay, so it looks very similar to E. coli. And Citrobacter frundi ferments the same sugars as E. coli. So positive, positive, negative, positive, with all the positives having gas bubbles. So usually the only difference between the two of those is the H2S production. Now, the last one that we're going to read is Pseudomonas Pioriginosa. So here's our TSI tube, and you will see that it is a red tube. So your result for Pseudomonas would be K over K, an all red tube. No gas produced, of course, because no fermentation occurred, and there is no H2S production, no black. So K over K, no gas, no H2S. And if you look at all four tubes, they're all negative. Now, the glucose, which is this one, looks a little orange, but it's like that because it started out that way. So here's a complete negative that doesn't have anything in it. Here's the Pseudomonas. So glucose starts out as more of an orange color instead of the red. Now, the Pseudomonas, you can see there's growth there. The bacteria grew, but it respired. If you can remember all the way back to the beginning of the semester, Pseudomonas is an aerobe. It's a strict aerobe. Remember when we did the motility test? It only stayed at the top of the tube. It's a strict aerobe. So it's only going to respire these sugars. Hopefully it's not confusing. It's way more straightforward than the selective and differential media.